Well, welcome everyone. Thank you for turning out this afternoon. Today's event kicks off a very special period of time in the life of the Cancer Center. This is the first of 5 days of celebration of the Cancer Center. And the uniting theme is Share the Journey. Now that's a metaphor that undoubtedly will have different meanings for each and every one of you in the audience. It has a special meaning for me, having had the honor of being the founding director of the Cancer Center. But regardless of whether you are a student, or whether you're a faculty member in the research laboratory, or if you're a faculty member who delivers clinical services to our patients, indeed there is one thing that unites us all, and that is our patients. And whatever we do, it is in the name of serving them better. And so it is that context that I think sharing the journey is especially significant. I will tell you that we have a treat in store for us today. I had the privilege of meeting today's speaker earlier in the afternoon, and I think you'll find it not only educational, but certainly inspirational given his exuberant personality. But it's also very special today in that we're honoring two very special people in the life of the Health Sciences Center, David and Susan Hardesty, and I know President McGraw is going to say more in just a moment. But I also want to take this opportunity, David and Susan, to personally thank you for all that you've done for the Health Sciences Center and the Cancer Center over all your years here with us, and so I want to do that in a very personal way in front of everyone. So I want to welcome to the podium President McGraw, who has been with us for a year and is going to be leaving us all too soon. But I want to take this opportunity also to thank Peter for his engagement in the life of the Health Sciences Center. As you know, we're talking about many aspects of change at the Health Sciences Center right now and reorganizing. Well, President McGraw very early on grasped the importance of this discussion and has been there for those of us in leadership roles, and he has personally met with many folks here at the Health Sciences Center. So Peter, thank you for sharing the journey as well.
Mr. Vice President, thank you very much. Ladies and gentlemen, I'm supposed to say, but I really mean it, it's a privilege and an honor to be standing here. It really is a privilege and an honor for many, many good reasons. The work we do here and the work that's done nationally to deal with the scourge of cancer and all of its manifestations is absolutely critical, and it is one of the fundamental obligations of any health science academic center, and it is one of our huge obligations here. And we are well led by Scott Remick and countless men and women who work with him. It is so important that we recognize today in this inaugural lecture David and Susan Hardesty. And it's professionally important. It's also personally meaningful to me. It's professionally important because David Hardesty served for 12 years as president of this exciting university. I've been in the president business a while too, as David knows, but I never made 12 years, David. I was able to hang on for 10, but 12 years is a long run. And it's much more than a long run in what David Hardesty has done. He provided strong, visionary leadership for this university in research, in teaching, in caring, and in showing in many specific programs that he implemented and nurtured. His caring for students, professional students, graduate students, undergraduate students. And he cared and he cares today. And he's extraordinarily active as a professor of law in our law school and as an advisor and a guide in many, many good ways to me, as he will be to my successor here as president, Jim Clement, who will be here in a couple of months. And David cares enormously about health and fighting cancer. So also for Susan Hardesty, a dear friend, both of them, really good friends. Susan contributed just as David did in the 12 years that she served as his hardworking and loyal partner on behalf of the educational work and the fight against cancer and good medical programs and caring about students. And Susan is the one who conceived, and it is one of the best programs in the country. She helped develop the Mountaineer Parents Club, an organization, not a paper organization, that provides parents a way to stay in touch with their students, with their daughters and sons as they're here. It's a thriving, powerful organization that actually helps us to attract and retain students who come here because they know this is a university that serves people, serves students, cares about all the things that a major university with a great medical center is all about. So when we talk about David Hardesty and his 12 years, we're talking equally, and we really are, about Susan and David's work together. They are my predecessors here, but they are much more than that. They're personal friends who I profoundly respect. And before I sit down, and we're going to hear a very great lecture by a very distinguished scientist who will be speaking to us and will be introduced by Scott in a minute, I do want David and Susan, this is a presidential order. I don't give a lot of them. I want you to stand up because we really want to give you a loud round of applause. Thank you, Scott. It's all yours.
Peter, that was perfect. So as David and Susan know, universities are big on tradition. So we start a new tradition in the Cancer Center with the inaugural Hardesty lectures. I got to know them at our my first gala a couple years ago and recognized the Cancer Center wanted to recognize their stewardship of the university with an endowed visiting professorship and lectureship. And I can think of no better person to be our first Hardesty speaker than Dr. DeRochelle. And looking at their respective CVs, there's a Harvard connection as well, so that's actually nice to see. Dr. DeRochelle, I can vouch, is a superman, because en route here, we lost his baggage from Washington, D.C. on a direct flight to Morgantown. His baggage showed up in my office about 2 o'clock, and he managed to change in the bathroom and get fresh set of clothes in time for the lecture. So he truly is a superman. He is the epitome of an academician, and I mean this in all sincerity. You don't hear it enough. He's a gentleman and a true scholar, and I think everyone who has met with him today has certainly appreciated that. He's the director of the NCI Division of Cancer Treatment and Diagnosis. He oversees a research team of over 800 scientists at the NCI devoted to cancer therapeutics and cancer discovery and development. He's an outstanding scientist, continues to run a laboratory at the NCI, and has throughout his career, with expertise in molecular and clinical pharmacology of a series of compounds we call anthracyclines, doxorubicin, which are actually the cornerstone of many cancer therapeutics even to this day, exploring the role of oxidant stress and signal transduction, and host of novel therapeutics for a variety of solid tumors in breast, GI, lung, and GYN cancer. He also led the first phase 0 trial, and I'll challenge folks in the audience. This is a wonderful concept that he and the NCI group have championed, and he's going to talk to us about it. He's the author of over 300 publications. He's a PI of numerous research grants and study sections. I've outlined his training here, Harvard graduate, on to NCI for subsequent training, and then to University of Southern California, City of Hope, where he was for a long, long time, joined the NCI in 2004. And it's good to see that he's adding West Virginia University to his list of special universities that he has been invited to speak at. Wake Forest University, University of Pittsburgh, City of Hope, Ohio State, and the New York Cancer Consortium and Albert Einstein College of Medicine. So it is an absolute distinct pleasure for me and a privilege to welcome Jim and to introduce him as our inaugural Hardesty speaker, David and Susan.
It's a great, great honor and pleasure to be here, and especially to be here to celebrate this wonderful week that you have ahead of you. And I was fortunate enough to get a tour of the new facilities. They are more than state-of-the-art. They will be wonderful additions to the venues that patients come for sometimes difficult therapies, and that environment is incredibly, incredibly important. And so I salute you for having made that investment. It will pay off for many, many years to come. Thank you for the chance to celebrate with you this wonderful week. It is very, very special for all involved. Scott asked me to talk about clinical trials. I'm going to do that. I'm going to give you some information and data. As my wife likes to say, if you don't show at least one gel, you haven't given a talk. So I will show you at least one gel with a little bit of molecular biology. But more importantly, try to give you my perspective on ways that we can do what we need to do for our patients. That is to speed up the delivery of new therapies to men and women all over the world. Just to give you some idea of the scope of the clinical trials program that the NCI supports, this is current as of a couple years ago. There are approximately 2,000 sites around the United States and overseas where clinical trials supported by the National Cancer Institute are ongoing. And I don't know if you can see in the back of the audience, but we put about somewhere between 25 and 30,000 patients a year on therapeutic trials at the NCI. And so it's a large enterprise. And in fact, if you combine the therapeutic trials that are supported by the government with the quality of life studies, the other kinds of interventions that are going on, the NCI is the largest sponsor of clinical research, cancer research in the world. And just to give you some sense that your tax dollars have been of use, these are trials and that report data that have changed practice from the initiation of treatment of CML with Gleevec to more recent studies with the use of angiogenesis inhibitors. These are all trials that our grantees have conducted that have led in one way or another to a change in the practice of medicine on a day-to-day basis for patients with malignancy. The real issue then, and what I'm going to focus on, is really this graph. And that is that to bring a new cancer drug to the clinic today still takes anywhere from 12 to 15 years. And that is if you're lucky. And that occurs at a cost of at least $1 billion or so. And the interesting thing about this is that the closer you get to the more definitive stages of therapy and the more definitive testing that goes on, the greater your risk. So you start out early on with studies that hopefully can get done easily, and this is what we want to avoid, obviously. But you start out with studies that we do, early phase studies, phase 1 so-called, and 2 trials, and we invest in those studies, but they're actually relatively cheap. And as we progress to larger studies that compare standard treatments to treatments that are investigational, the costs dramatically skyrocket, and yet at the same time, the failure rate increases in proportion. So the closer we get to doing something that might actually affect clinical practice, the larger the chance that as we get to late phase testing, those drugs will turn out not to be useful, mostly because, not because they're too toxic, but mostly because they're just not effective enough. And so it is more than a tragedy to spend 15 years going from the initiation of experiments and then experiments in humans to the point where you have to give up on an agent because despite all of the science, despite all of the testing, and despite hundreds and hundreds of millions, maybe billions of dollars, those drugs don't work. And so our job, really I see it as our job, and I include all of you in this audience, is to think about how we can do this better. And this is a very simple diagram, but unfortunately, as we stand here and sit here today, this is still pretty much how we do clinical cancer trials. We take information from the laboratory, we do some preliminary testing in animals, using models, that's a subject of a whole nother lecture of mine, that are really not very good. We make sure that we can do this relatively safely, and then we do small initial trials. We then try to use a larger number of patients to find out specifically where a particular drug might work. And then we need very large numbers of patients. So we go up an order of magnitude as we progress through this process in terms of the number of patients required, and yet these studies often fail. And so one of the things that I've been most interested in the 5 years since I moved from Los Angeles to Washington, D.C., was to try to think about ways that we might be able to improve this process. And I say this with all due respect to my great friend and colleague, Meryl Agorin. If we still tried to measure drug levels the way we did in 1970, and I had one of these machines, something very much like that, you can see the level of effectiveness. I mean, that's an amount of drug, that particular drug that I've been interested in, that Scott mentioned, doxorubicin, that if it were in your bloodstream, you would die. You would die almost immediately. And yet that was the limit of sensitivity of measurement. We got a little better in the 70s. I remember great excitement in getting my first HPLC, and we got down into the micromolar range of drugs, still way higher concentrations than ever really reached tumors or are useful. And it's only in the last 5-10 years that we have the kind of instrumentation that actually measures drug levels that are pertinent to the kinds of levels that are required to kill tumor cells. And yet we would no more today use one of these instruments to measure a drug level in a patient than it would be. These are in museums. Whereas we still use the same clinical trials mechanism that started in the early 1960s. We still use that methodology today to test our drugs. And clearly, we have to match the new technologies that we have available to study new agents with the kinds of clinical trial designs that will be more useful. And so what we started thinking about and trying to utilize is something that is much more intensive but much more limited. And that is to say, as we know more and more about how these new drugs work, can we conceive of, can we develop, can we utilize the kinds of testing that will allow us to take 5 patients, 10 patients, 15 patients, study them in very, very intensively and repetitively as needed, to test specific hypotheses, to have the molecular assays and find out whether or not a particular drug, which we have a better idea how it might work, does it actually hit its target? Can we demonstrate proof of principle, something that in 15, 20, 30 years ago was simply not possible to conceive of because we had no clear idea how particular drugs actually worked. And as Scott said, I've spent a better part of 30 years of my career trying to study one particular drug. And I remember my father died about 6-7 years ago, but he was a retired pediatrician, and I used to come home and he'd ask me, "What are you working on?" And I said, I'm still trying to figure out how adriamycin works. He says, "You've been doing that for 25 years. Haven't you figured out by now?" He couldn't understand that you could work on something and something could be so complex that you couldn't understand how a particular molecule might work. But that was the era of using drugs, using molecules that were broadly pleiotropic in how they affected tissues, normal tissues, cancerous tissues. And it's not a surprise that many of those agents introduced in the 60s and 70s, though effective, we still do not know definitively how they kill tumor cells. It's a new era. And so when we develop agents now, we have a much better idea how they might work, at least initially. And so we have to think about, in the earliest phase of our clinical trials, developing the targets and trying to measure specifically the effect of those particular molecules on the targets in tumor. And we need to be able to do that in relatively smaller trials where we invest a lot more upfront. And the whole rest of my talk is going to focus on how you can think about investing relatively more, but not billions of dollars more, earlier in the trial process to do what really needs to be done, and that is to fail fast and fail early. It's much more effective to spend more money and perhaps $5 million, $10 million, not $1 billion, to fail a molecule and never get to the point that you're using something that is ineffective in a patient. And so what we've spent much of the last 5 years on, and I'll show you, are how to basically utilize an intensive program of animal-based research that will allow proof of principle investigations. So we were, and I have to give credit because I would love to take credit for this, but it's simply not mine to take credit for. This is an effort that began before I moved to the NCI and intensively involved colleagues of mine at the National Cancer Institute working with the U.S. Food and Drug Administration to figure out a way to change regulation so that you could be able, with less of an enormous investment in the kinds of research, the toxicologic research, other things that are very expensive, and so that you could get things into patients very early, find out if they're useful or not, and then do a full-bore development plan. And so the so-called exploratory IND was promulgated in January of 2006. The work for this began early in 2003, and I'm going to talk about what this means and the actual completion of the first cancer clinical trial utilizing this particular FDA guidance. So what does this allow you to do, and why is this of some importance? Well, this is the standard preclinical development pathway that one has to go through for any new small molecule. And perhaps the most important part of this is that one has to do very extensive toxicological evaluations, appropriately so, for patients. Patient safety is always paramount, but it's unclear in this day and age with the kinds of molecules we're now developing, which are very, very potent and relatively non-toxic. That is to say, they're much more highly targeted than the kinds of broad cytotoxic effects that we used in the past. And the issue regarding this particular FDA guidance was, could you think of a way forward, an investigational path in which you would use less toxicological investigation? These kinds of studies can cost up to $1 million for a single molecule, sometimes more. And so even thinking about going through this entire development path is very daunting. Could you do less safely that would allow you to get a drug into patients and find out whether you can actually learn what you need to learn, whether or not you hit the target? And so this guidance basically allows one to do three things more easily than had been done before. With substantially less toxicological evaluation, one can do bioavailability studies, basically pharmacokinetic studies to find out whether a new molecule, and most of the new molecules we develop today are oral molecules. Patients don't have to come and sit in the hospital or spend hours getting these drugs. But the key thing in their development is to find out, are they absorbed or not? And so the FDA recognized this, and it's now possible, given the very, very high-powered instrumentation that's available, to actually test in a patient whether or not a very small dose, given orally, a dose that is very unlikely to have a lot of side effects, actually reaches the bloodstream. And that was not possible 15 years ago because the instrumentation, the sensitivity, was not possible. Why is that important? Well, it's important because the standard way that one develops drugs, one comes through a very prolonged process, but usually with several possibilities, several analogs that one might be a little better in its solubility, one might be a little more potent, one might be a little easier to manufacture, but it's unclear whether any of them is well absorbed or whether they hit their target. And so the availability of this investigational IND process allows one to very rapidly, in a small number of patients, find out, does the drug get absorbed, period, end of case. The ones that are not absorbed, and you cannot tell this from an animal model, even a large animal model, only can be done in patients, so that in the space of a month, one could lessen the burden of testing by narrowing down the focus of the drugs that you're going to study. Another very important ability that this new regulation allows is allows us to do imaging studies. And this, of course, is the future of oncology, we're going to be, as we develop new compounds, we're going to be simultaneously developing them as imaging agents and therapeutic agents. Does the drug get to where it's supposed to go? And the ability to do this and to do this quickly is really a major advantage of this process. And what I'm going to spend my time on is the third possibility that this new guidance allows, and that is to do relatively short, less than 1 week exposures of a new drug to a patient with cancer for the specific purpose of finding out whether there's a change in his or her patient's tumor that reflects the perceived mechanism of action of that compound. So what are the differences between the paradigm that we've been following for the last 40 years and this new paradigm? Well, in the paradigm that was developed for the study of cytotoxic molecules, the primary endpoint was to establish how much drug you could give. I mean, it was a safety study, and this is the overall a major point. The point of these pilot studies, so-called phase 0 studies, is to find out, do you modulate the target? Okay? And inherent in that, therefore, is that you have to have the appropriate kind of assay to measure the target. So what does that mean? Well, you need to have a pretty darn good idea what the target is, number one. And number two, you have to invest in, before you know this is going to work or not, the development of methodologies that are sufficiently robust so that you can make conclusions from relatively small numbers of patients. So the key feature of these trials is what we like to call this transition from preclinical to the clinical arena. You have to have developed these very robust assays. You have to demonstrate that there's a relationship in an animal between how much you give and whether if the target is inhibited, does the tumor shrink? Those are things that are not always done in standard drug development paradigms. And you need very innovative ways and robustness in your assays to allow you to do these studies in small numbers of patients. So what does this get you? So it gets you entree into this fail fast, fail early paradigm, but it also allows you, if this type of study is effective, it allows you to find out whether or not the assay you're developing for the marker of interest of the molecular target for the drug, does it actually work? And I say this with all humility. I spent many years trying to do these kinds of studies before this paradigm came into fore, thinking that I could do something in my laboratory that worked great in a tissue culture dish and immediately go and apply that assay to a patient. And it is a recipe for disaster. It simply doesn't work, mainly because what is perfectly reasonable and what is more than sufficient to allow you to get a grant from the government to study something that works in a tissue culture system is simply almost never robust enough to use in a very small sample from a patient. And so you really have to demonstrate that what you're doing is applicable in a clinical situation. And to do that, unfortunately, fortunately or unfortunately, depending, requires a lot of resources. And it requires a coordinated development of assays in animals. It requires a team that learns how and spends the time learning how to handle tissues and to get things from a radiology suite to a special assay laboratory. And it takes an investment in understanding how to acquire the tissues so that they are suitable for the biomarker or target that you're trying to assess. Let me just, as an aside, it's related, but just to make the point, and I think those in the audience who actually work with experimental animal models of cancer, and unfortunately would probably fess up to the following. We, of course, have rules about how animals can be handled. That's fine. But I never thought twice about, once the animal was euthanized, how to resect the tumor. Were there issues about how fast it had to be frozen? Were there issues about handling it, its size? Did it make a difference? The animal was sacrificed. I cut out the tumor, went on about my business in my laboratory. Well, it turns out that, and these are issues, the reason nobody thinks about this is that it's essentially impossible to get a grant to study this. So if you sent a grant to my division that said, I want to really study how to develop an assay for and to handle the material necessary to answer this question, the grant would get an unfundable score, no hypothesis. No hypothesis. Well, here's some data. I'll show you some data. If you don't do this, in essence, demonstrate that the assay that you want to develop is clinically ready, that you know how to store it, you know the time frames necessary, you know how stable something is if you freeze it and thaw it and freeze it and thaw it. You do all of these things, and if in fact you can't get it from the radiology suite to the laboratory that you're doing it without destroying the sample. Now, I'm sure the same thing occurs here as occurs in Bethesda. The NIH campus has lots of big buildings, and none of them are connected by tunnels. right? So in the summer, you have to take a piece of tissue and literally run from the hospital to another lab building. And it can be, well, you know, Bethesda, it can be 100 degrees, 95 degrees. Does that sample going to survive, you know, a 20-minute walk in an ice bucket or not? Well, you have to find these things out, but no one will pay you to do it, and no one will pay you to use something that is used every day in the CT suite in radiology, an 18-gauge needle biopsy, and find out in an animal, can you make all these things happen in a way that will transfer immediately to the clinic. So we did a series of experiments, actually a very large series of experiments, to find out if you could do some of these things. And this is just one set of experiments in which we took basically, this is a human melanoma growing in a nude mouse, an athymic immunocompromised mouse. And here we have a standard needle, and we do the biopsy of the anesthetized mouse. Here after we did the needle biopsy, we cut out the tumor, right? And then we used a device that is commercially available. It's used every day in the radiology department when one does ultrasound-guided breast biopsies. And it's a device that basically, first the device goes in and freezes the tumor in situ, and then cuts the piece off. So it's frozen in place, and the notion being, well, what's the question? The question is whether or not freezing the tumor in place actually does it have any effect on the targets we're interested in measuring. Because some targets, like RNA, like the presence or absence of phosphate groups on particular target molecules, are very, very labile. And here's the evidence. If you resect the tumor, this is phosphorylated AKT, an important target for a variety of different drug development programs. If you cut it out, which you would have thought, you get the biggest piece of tumor, it's frozen immediately after resection by a veterinary pathologist, done as quickly as is humanly possible. Well, there's almost none of this phosphorylated protein left. If you, on the other hand, if you freeze it in place and then cut it out, if I showed you this and relabeled it, you might think that in this line we had several very important inhibitors of this particular phosphorylated protein. But in fact, it has nothing to, these animals got nothing other than the handling of the tumor was done differently. And so we did a series of experiments like this one, but for an enzyme that we were interested in, poly-ADP ribose polymerase, a critical enzyme that is necessary every day for you to repair your DNA, right? For which we developed an immunoassay, very, very sensitive immunoassay, and we wanted to know, well, what if we do these very simple kinds of experiments? What's the difference between doing a biopsy with a needle and cutting out the tumor? Is there any difference? Well, it turns out that it's actually better and you get higher levels of this particular enzyme with no treatment, no difference in handling other than excise it or put a needle in it and take it out. You get higher levels if you take a smaller piece. Well, why is that? Because it freezes more quickly. It freezes more quickly. Not rocket science, okay? But the fact is that these kinds of experiments like this one, where we said, okay, is there a difference? If we put tumors of different size, right side, left side, big tumor, little tumor, is there any difference? It turns out that there is no difference. If we take one piece now and we wait a week and we biopsy again, which often happens in our patients, well, is there any difference if you do that? Do you perturb the environment of the cancer? Well, not in this particular model. And we went through a very elaborate set of things that are totally mundane, right? But to the best of my knowledge, had never been systematically evaluated. And this is only one tumor and one target, right? But we, by doing this, understood that, had a much better, we thought we understood that if we went into patients, we would be able to do things in a much more systematic way. In the same way, we could show that if we did a time course, if we gave more drug, okay, this is more drug, do we get less and less of the enzyme? Yes. But then what happens? Well, if you give a dose and 2 hours later, you get the peak inhibition of this enzyme. If you allow 5 hours to intervene between the single dose, it starts to recover. by 24 hours. It's not completely recovered, but it's partially recovered. So we had a sense of how long the drug actually worked given once, just one time, to a mouse. So when we are thinking about doing this, and I'll show you some specific application of those results to our patient trial, this figure gives you kind of an overall set of guidelines in terms of how you figure out whether or not the target and the drug are amenable to these intensive, small patient studies. And basically what it boils down to is the following. If you have a compound and if you have a target that you can deliver at a non-toxic level and the variability of your target is low and you get inhibition at a non-toxic level, you can do these pilot studies. Even if the variability is high, but you still can get a significant decrease at a non-toxic level, you can do something like I'm going to describe. On the other hand, if you have a target that is such that only at a toxic dose can you perturb the target sufficiently to be sure that you've made a biochemical effect, it's unlikely that these kinds of studies are going to be very useful. So I'm going to describe now a clinical trial. And first I have to say something about the team that conducted this trial. It's a lot of people. I guess on the one hand, it's a good thing I work for the government because we had a lot of people that we could throw at this project. But most importantly is not the number, but rather the interrelationship of these people. So we didn't just need physicians, we needed dedicated radiologists who could do these biopsies. We needed people who developed the assays. We needed people whose sole job it was to get from a radiology suite to the laboratory where these were done. And we needed regulatory people and so on and so forth. So I want to make sure, I'll say it at the end, but I want to make sure at the beginning I say that this is simply not my work. This is the work of a large number of individuals who work together across an entirely, a very broad multidisciplinary landscape that allowed us to do these studies. The last thing I'll say in preparation is that we really had to invent a new kind of statistics because, anybody knows how dumb could you be to try to do a study with 10 or 12 patients. Everybody knows bigger is better. Well, the whole point is whether we could do smaller is smarter. And the point here is that you can do these things, but as I've probably bored you to tears with, you can only do it if what you're measuring can be done with a very narrow amount of error. Okay, last but not least, a phase 0 study, so-called, or a study conducted under the guise of the FDA's investigational exploratory IND, is a study that is done without therapeutic intent. Either it's an imaging study that has no therapeutic intent, it's a bioavailability study where you give a drug to a patient by mouth and find out how much is absorbed one time where there's no potential for therapy, or it's a study where we're trying to understand whether or not the target has been altered with a single dose of drug. And so it is very, very important that we spent a considerable amount of time with our ethics group at the NIH trying to develop a process to obtain truly informed consent for these procedures. It may at first sound, well, how do they do that? That couldn't be the right thing to do. Well, but one thing I have to tell you is that it actually took a lot less of, it was a lot less of a problem for our ethicists than I at first thought, because when it became clear to them that we were giving patients doses of drug that had an extremely low likelihood of having any toxic effect, they actually thought that these kinds of pilot studies were easier to justify on an ethical basis than a standard phase 1 trial where we have a small chance of benefit, but it is very small, but there is a reasonably good chance of toxic side effects. So this is actually something, and since patients are they're enrolled in these studies for a day or 2 days, and we can put them on other investigations in a relatively short period of time. We actually had no trouble accruing patients to these trials, but it does take a significant amount of time and effort to document that the fact that there is no intrinsic therapeutic benefit is part of the process. So the objectives of the trial that I talked about and I'm going to present to you are really to determine whether a single dose of this drug called ABT-888, it's made by Abbott Oncology, and is an inhibitor of this DNA repair enzyme, poly-ADP ribose polymerase, actually did it hit its target? And how much drug could we find after a single dose in the patient's blood? And the whole trial was done with 13 patients. And as you'll see in a minute, we probably could have done it with 10, but we decided to do some additional studies in terms of recovery of the enzyme that added the additional 3 patients. These are patients who all had many other forms of investigational therapies. They'd had lots of other treatment and had a variety of different kinds of malignancies. Here's the study schema. It's really fairly straightforward. That is to say, initially, all we did was to get to blood and to measure this enzyme in blood, then gave a single dose, and then collected blood for the estimation of the amount of drug in the blood of the patients. Our criteria for going beyond this were a particular level that we had to achieve in the bloodstream that we knew was effective in animals. And at that point, the study was designed so that as soon as that level was achieved, we would then do biopsies in patient tumor samples to understand whether we were hitting the target. So, as in every area of medicine and science, it's probably better to be lucky than smart. Our first patient who got the first dose of this drug hit the target that we were aiming for. So we knew that even at the low dose we started at, we should be able to see some biological effect. And we had very straightforward absorption of this drug orally with essentially no side effects, a single dose. And then this really is the take-home bottom line. We found that as we increased the dose in the peripheral blood circulating mononuclear cells, for most patients, we got a very significant inhibition of the target enzyme. And as we started to measure inhibition of the target enzyme in patients' tumors, in most patients, 5 out of 6, we had inhibited by over 90% the amount of enzyme at the time course, that is 3 or 4 hours after a dose, that our animal studies had predicted was the point in which we would see the maximal inhibition of this particular repair enzyme. The 3 additional patients that were treated are shown here. They were dosed at the highest dose, and then we obtained biopsies 24 hours later. And what this shows is that in some patients, that inhibition goes on for 24 hours. In some, there's recovery to up to 50% of a dose. And so this helped us because in future studies, it became clear that this drug could be given once a day and still produce a very significant inhibition of its target. This just shows that there is a very good correlation, and this doesn't happen very often, between the inhibition in the tumor and the inhibition in a surrogate tissue, in circulating blood cells. So why is that important? Well, it's important for several reasons. Number one, because it doesn't happen very often. But more importantly, number two, if this is real, and I think it is, we have a lot more data than I'm showing on this slide, it may well be possible to, in future patients, get a very good estimate of what kind of anti-tumor effect on their target is produced just by getting a tube of blood, rather than putting patient through a biopsy procedure that has substantive risk. Getting to the end, and I think I'll be on time, I wanted to give you a time course for the study overall, because the most frequent, probably the most important question, and the question that I started out with, does this actually save time? Is it going to bring drugs to patients more effectively? And so what you see is that we submitted an exploratory IND in the middle of April of 2006, and basically in about 60 days, we were allowed to proceed by the FDA. First patient was put on, and literally within 5 months, because by the time we had done the second group of 3 patients, we already knew that we could get 90% inhibition of the target. And so we finished the trial, and you've seen the data, but what does this actually allow us to do? Okay? Well, it allowed us to work with the pharmaceutical company to skip the standard phase 1 approach to this drug. they knew, we knew, the biochemists knew, that this is most likely not to be a very effective drug by itself, but is very likely to enhance the ability of other drugs that damage DNA to enhance their therapeutic effectiveness. And so, because we knew what dose and over what time course one could inhibit the target of interest, the company went immediately to designing combination trials of this agent with other drugs that damage DNA. And what I put down here is just to give you some sense. So it took about a year from the start, the first patient on this trial, to the start of combinations of targeted agents, which is where this field is moving. If we look back over a prior 10-year period, 90 prior trials in which the National Cancer Institute held the FDA investigational new drug application, how long did it take from first-in-human single agent to first-in-human combination agent? 900 days. So we'll see. The answer is we'll see. It's very optimistic, but I think that it really, for this particular drug, was a process that helped, and certainly, even more importantly than the time, it helped think about how one should structure these additional trials, which will be most important for how this drug, if this or other drugs of its class, get used in patients. So when do you think about this approach? Well, this is actually relatively straightforward. If you have no assay for your biomarker, this is not something you're going to do. If you have no idea how the drug works, again, you're not going to be able to develop an assay, so this approach is not appropriate. If the compound has a very narrow therapeutic index and has to be given intravenously, again, it's likely you're going to have to do a standard safety trial. But even then, developing information about the mechanism of action of the drug is likely in the early phases, if you invest in it, to help you plan better for the future development of the agent. So what we've written about is basically a coordinated process where you look and do these pilot studies, and then they inform a much more lucid, targeted way to do the formal additional studies that bring the agent along a drug development pipeline. To make the point, perhaps for essentially the last time, here's the traditional approach to phase 1 studies. And you do all of these wonderful things, which take a big team to do, but in fact, until you're at the end of your phase 1 trial, you can't make a decision whether you're going on because you don't know, are there untoward toxicities that you hadn't expected? Is it really acceptably toxic, and so on and so forth? This process allows you to make a decision much earlier because you get much more information, especially if, for example, in a bioavailability trial, if the drug doesn't get absorbed, the drug is dead. And you learn that with a very small number of patients. So our hope is that going forward, we will replace this model, which is Model T as being kind, to a model in which we spend a lot more time developing our understanding at the preclinical level so that we can do much more targeted and smaller, earlier phase studies that potentially may even obviate the need for these large studies that are now required for FDA approval. Here's our paradigm in 1989. I think there is a different paradigm, or at least one can think about a different paradigm, in which one can look very broadly at the kinds of patients that one wants to enroll on clinical trials that are not so much guided by the type where the cancer started from, but what is its molecular phenotype? What are the molecular characteristics of the tumor that I think overwhelmingly going in the next 10, 15 years will be how we focus our trials. They'll be much, much more specific. And I hope that will allow us to spend 5 or 6 years, not 15 years, going from the discovery of an agent to FDA approval. So I could not end without thanking this large group of individuals at the NCI without whom this whole process and certainly this talk would not have been possible. And thank you again for this wonderful honor to come here and for this wonderful, wonderful week that you're about to embark on. Thank you so much.
Any questions for Jim? Tom?
How will what you said impact or relate to the traditional 60-cell screening panel of the NCI?
So that is one discovery tool that we have, okay? But what we've tried to do, and are in the midst of doing, is to try to take that tool and to make it much more molecular. So yeah, I don't know how much you know about that panel. It was started, as in so many things, and if you're old enough, everything is old is new. In the early to mid 1980s, my mentor, former mentor at the NCI, decided that animal model testing with these nonspecific mouse tumors was useless and it was not going to get us anywhere. And surely if we put together a large panel of human tumor cells, we could do all of this testing in vitro and we would learn much faster, and we could do, and whatever, if we had 10 colon cancer cell lines and 8 breast cancer cell lines, that would direct us to what kind of drugs would work in what kind of tumor. That doesn't work at all. It's a complete failure. Okay? What is not a failure, however, is that the pattern of inhibition of the growth of those cells is actually relatively unique for different classes of molecules. And so what you can do is to take, and over 30,000 compounds have been screened in that way. And so if a chemist discovers something that he or she thinks is novel, it can be tested, and one has a computer algorithm to actually compare the pattern of growth inhibition in those cells of this large number of prior molecules to something that is new. So that's all done now based on simple growth assays. And what we're trying to do is to actually move into the 21st century and try to understand, with especially our targeted agents, what is the profile of genomic changes that occur in those cell lines after they've been treated at concentrations that are likely to have important biological effects. And this may seem, please, I don't mean this to be sound like it's hubris. It's really built out of frustration, but I truly believe that the characterization and the categorization of the antineoplastic drugs that we use today and what you teach your fellows, okay, is probably well nigh unto useless in terms of how drugs actually work. And when we develop a molecular understanding of what genes are altered, which we simply don't know, even for the oldest drugs, it's not known in a comprehensive way. It's not known in vitro. It's certainly not known in animals. It's likely that the table of contents of cancer pharmacology journals will change, because what we think or we thought originally were the targets for these agents turn out to be way too simplistic, that there are patterns of molecular pathways that are involved, and that various agents of different chemical classes will fit together under these pathways. That requires very high-level computing power. It requires approaches that are only now coming to the fore, and so we're trying to utilize those models to really relearn an ontology of cancer chemotherapy.
Tom?
Yeah, I wanted to ask you a question about the cellular compartment that you assayed for your target biomarker. Now, it seemed to me, one of your middle slides, that what you did is you were assaying the mononuclear cells in blood.
That's one of the things that we assay.
Okay, so, and that's where my question was. Now, are these mononuclear macrophages? Are there other mononuclear cells? Did you cell sort them? What cell compartment?
No, these are non-granulocytes.
Non-granular.
Non-granulocytes.
Okay, now, so the reason I asked that question is that since you showed, whatever, 19 patients or 20 patients in this study, and they had
Only 13.
or 13, okay, and they had different type of malignant involvements, were those still going to be the common cellular compartments that would be relevant to all those tumor models, or is there different cellular compartments, biomarkers for different tumor cells?
So it doesn't appear so, okay? So the two things, how can I comment? We have tested this particular agent in a relatively large, what does that mean? A dozen different human tumor xenografts, okay, of different basic human histology, melanoma, colon cancer, so, okay. It is a very potent inhibitor of this enzyme that is present in all of those different histologies, and it's so potent that it inhibits in all of those histologies by 95, 100% the enzymatic activity of ADP ribose polymerase in tumor.
And you showed the correlation because it correlated with tumor types on the biopsy using the peripheral mononuclear cells as your assay. So now you don't have to do biopsies. So that was the point of your slide.
Mononuclear cells, the carrier of this?
So,
If they invade the tissue compartment?
No. So this may well be one of those classic examples of true and related, okay? Because most of the time, I would say 95% of the time, when we look for a quote-unquote normal tissue to reflect what goes on in tumor, and we use peripheral blood, we are sorely disappointed because almost all of the time, it's a waste of time. And so this was very surprising, and it's got to be something that is genetic because we had one patient out of this series of 13 whose tumor biopsy showed minimal inhibition by the drug and whose peripheral blood cells showed minimal inhibition of the drug. And when we got that patient back, and that was true when the drug was administered systemically or orally, okay? We got the patient back and got more peripheral blood mononuclear cells and tested in vitro exposure to the patient's own, still could not get inhibition of this target in that patient. Looked for SNPs, looked for a variety of different features of that individual's enzyme, and could not divine what it was that made that individual resistant. But there appears to be a relationship, the nature of which I simply cannot stand up here and tell you because I don't know, that the activity of this inhibitor in peripheral blood cells is related, not one-to-one, but linearly to the levels and the effectiveness of the drug in the tumor. But why, I can't tell you.
Ramin.
So what is the, now we have in the era of the genomics and genetic signatures of tumors, and we see not all the lung cancer have the same genetic background. But the future of the phase 3 trials, randomized, double-blinded clinical trials, how are we going to be incorporating the knowledge of genetic signatures to those big studies?
So as Scott and I talked about, I mean, there are really two things that are going to happen. One that's already happened, in part at another institution that I won't name, but one that is going to be facilitated by a new NCI core facility, and that is that I think we are soon going to be in the era in which every patient will have a piece of his or her tumor characterized over a wide range of genomic and other molecular pathways. And that will happen whether or not that individual needs treatment. It may happen, they may be cured by their surgery or may not, but that information will be available for use subsequently in clinical trials. That it may be that you have a patient, let's take the circumstance for C225, for cetuximab 5 years ago. You didn't know that KRAS mutation was important. I didn't know, okay? My division was paying millions of dollars for trials with that antibody and had no clue that half the patients were being treated weren't going to benefit, right? But if every patient entered in that study had tumor, fresh tumor, or tumor that was available from a block was available for sequencing, you had done that a priori, just because you think that RAF is, it's an important, these pathways are interrelated, and you would have done that, you would most likely have had information far in advance of the clinical information, that there were patterns, right? Or subsequently, you might do in an adjuvant study, you might characterize patients with stage B colon cancer, who now it's pretty controversial about whether or not they benefit from chemotherapy or not. But it might well be that 5 years from now, if you have 1,000 patients characterized, they've all had their tumors characterized in a variety of different ways, you will then be able to call up a data bank of patients, and rather than having the patients wait for the patients to come to your office to enter into your trial, you will consult a bioinformatic database and find patients all over the United States that will be asked to participate in the study based on the characteristics of their tumor. That's what I think is going to happen.
Let me ask one question, and I'll pick your brain after too as well. Two things, I guess. You left out a lot of the toxicology stuff. Was that being done concurrently once you got beyond your phase 0? Because you talked about how that was being sliced out.
Okay.
So I don't know if that was being concurrent.
So a limited amount of toxicology was done in advance, okay? And so also what happened, and on that slide, that timeline slide, again, we were working with a for-profit entity. So these are smart people. So as soon as the second cohort of patients, they said, oh, this is a drug we're going to take forward. So now we're going to invest another $500,000, $1 million in the appropriate toxicology, and we're going to invest it well. And I can tell you from my conversations with those individuals, their ability to get resources from their company, having this data available, is much easier, because who's not going to bet on something that had much higher likelihood of being useful?
Now, you're ready for your phase 1 combination in a year, and the ethics of this are very challenging, not a topic for here, but are any of your patients able now who maybe had real marked depletion of PARP eligible to go on your combination trial?
So yes, with several patients who went on this initial phase 0 trial, actually were enrolled a few months later on our phase 1 trial of this drug plus topotecan.
I was intrigued by your idea of going right to the combination trials. And one of the issues you talked about in phase 0 trials was that perhaps drugs with a low therapeutic index versus a high therapeutic index and selecting out from phase 0. It seems like most of the drugs we have are kind of in between sometimes. They might have a cardiac toxicity, but they're not a traditional cytotoxic drug. How does that weed out? Is that still going to be susceptible?
I think it depends whether it's a chronic or an acute toxicity, right? So, you know, I mean, it's so fine. I mean, Gleevec cardiotoxicity was not apparent on early studies or short duration of exposure studies. So those things, I think, will only sort out when you treat larger patient populations. And I don't think they have any meaningful impact in your ability not to be able to do these kinds of early studies to weed out things that don't hit their target, so therefore you're not interested in them.
Fred?
I'd be interested in your perspective, enrolling patients, and many times as you allude to the bioethical issues, and generally enrolling patients when you can promise the hope of therapeutic value. But in this case, you went to great lengths from an ethical standpoint to indicate there was no expectation of therapeutic value. I'd be interested in knowing what are the personal motivations.
Okay, well, so two things. Number one, I mean, it's easier, I think, to think about if you think about a phase 0 trial as a normal volunteer study in a cancer patient. Okay, so what are the I mean, normal volunteers go do this every day for pharmacokinetic analyses, et cetera, except that we must have to do this a patient who has a tumor that we can characterize, right? So we ask people, so it's basically altruistic. Overwhelmingly, it is a tremendous sense of altruism. And it's also true that many of these patients, again, for those in the audience who do a lot of investigational therapy, when you have patients who are coming to a phase 1, an early phase, they've been through all of the usual treatments. These are folks who are really interested in not only their own welfare, but the welfare of others. And they will tell you time and time again, you know, if this helps somebody else, I'm going to enroll in this. And that's the dominant motivation.
This has just been a terrific kickoff to our week, Jim. And the questions, we have a reception, but before, we just want to give you a small token.
It's
of our
under $20.
It's
right?
under $20. It's under $20, so we'll let him open this in the back. And thank you, everybody, for the inaugural Hardesty lecture. We have a reception in the back here.